Hi, my name is Iram Wahid, and today I would like to share my research about controlling the sperm speed by activating toll-like receptors 7 and 8, which I had the privilege of working with Dr. Mehdi in fall of 2021. This research takes advantage of the functional differences between X and Y spermatozoa that are masked during fertilization to separate the sperms into two distinct populations. Male and the female reproductive cells are called gametes. Tested produces a male gamete, sperm, which are either X or Y chromosomes. Ovaries produces a female gamete, ova or X cells that comprise of X chromosomes only. Reproduction process of human. Oogenesis in the ovary results in the egg or the ovum with haploid number of chromosomes. Spermatogenesis in the seminal vesicles and the testes results in sperms with haploid number of chromosomes. When the sperm fertilizes the ovum, the resultant zygote has diploid number of chromosomes, which undergo mitosis to produce embryo with diploid number of chromosomes. Determination of the biological sex. The combination of sex chromosomes determines the biological sex. If X spermatozoa combines with the mother's Y chromosome, the offspring is a female. If a Y spermatozoa fertilizes the mother's oocyte, the offspring is a male. Probability of having a male or female offspring. Fertilization is a random process with equal probability of having male or female offspring. This is because the spermatogenesis in the mammals follow Mendelian segregation with 50-50 ratio of X and Y sperms. Environment in the female reproductive tract also affects the viability of X and Y spermatozoa. Differences between X and Y sperms. Differences may exist in two areas, the different chromosomes and different chromosome, that is the differences in the number and the kind of genes, and different sperm structure and function, that is the different expression of the genes. X and Y spermatids express different genes responsible for proteomic differences. Sperm screening methods. Recently, flow cytometry using HUCAS 33342 dye is used by the farming industry. Modified swim-up technique using RT-PCR is also used in the farming industry. Immunological approach using biomarkers, surface antigens, or antibodies is currently under investigation. Disadvantages include high economic costs, sperm damage, and low pregnancy rate. Scientists need to develop more specific, non-invasive, and cost-efficient approach. Recent approaches to reduce constraint. Genomic and proteomic studies offer a breakthrough approach for semen sexing, such as differential protein profiling of expressed proteins, using sperm surface proteins as potential biomarkers, and using toll-like receptors 7 and 8 as a potential biomarker for separation of X and Y sperms. Advantages of proteomic-based approach. Proteomic-based approach are more specific and sophisticated, and they're more precise. They result in less damage to the sex sperm, thus increasing fertility. Bulk production of sex semen with minimum cost. No special equipment or professional skills are required, and they can be done easily in IVF labs. Toll-like receptor 7 and 8 as a potential biomarker. Toll-like receptor 7 and 8 encoding X chromosome is expressed by 50% of round spermatodes. Toll-like receptor 7-8 ligand activation suppresses the mobility of X sperm selectively. Toll-like receptor 7 and 8 have common ligand resiquimod, which binds to toll-like receptor 7 and 8, and imicomod R837 that is specific for toll-like receptor 7 only. What are toll-like receptors? Toll-like receptors are type 1 transmembrane receptors known as pattern recognition receptors. They are present on dendritic cells, macrophages, and leukocytes, and they play a key role in the activation of innate immune system. Toll-like receptor 1 to 10 are present in humans and 13 in mice. Toll-like receptor structure. Despite a wide variety of ligands recognized by the toll-like receptor, they share a common structural framework in the extracellular domain, which consists of leucine-rich repeats. Then there is a transmembrane helix that spans the phospholipid bilayer and the intracellular toll interleukin receptor, which is present in the cytoplasm. Toll-like receptor family. Toll-like receptor 1, 2, 4, 5, and 6 are present on the plasma membrane, and they recognize the bacterial lipopolysaccharides, peptidoglycan, gram-negative lipotecoic acids, and the zymosan of the yeast and the parasites. Toll-like receptor 3 is present in the endosome and recognize the double-stranded RNA virus. Toll-like receptor 7 and 8 is also present in the endosome and recognize the single-stranded RNA virus. Toll-like receptor 9 recognizes the CpG DNA of the bacterium. Toll-like receptor signaling mechanism. The toll-like receptor extracellular domain, which consists of leucine-rich repeats, recognizes the microbial signatures known as pattern-associated molecular patterns or the damage-associated molecular patterns. Toll interleukin receptor recruits adapter proteins such as myeloid differentiation protein 88 and drift protein. This results in the activation of the cascades of reaction, resulting in the activation of MAP kinase pathway, nuclear factor kappa beta pathway, and interferon pathway, which results in the production of pro-inflammatory cytokines and chemokines. Purpose of this research. This research takes advantage of proteomics and genomics data to separate sperms into two different populations. This improves the quality and the quantity of production in livestock industry and allows for the preconceptual gender selection to the families longing for a specific gender. Effect of toll-like receptor 7-8 agonist on the ATP production. Toll-like receptor 7-8 agonist binds to its receptor on the X sperm, which are present on the tail of the X sperm. When the agonist resiquimod binds to its receptor, it results in the phosphorylation of the nuclear factor kappa beta and the glycogen synthase kinase alpha beta, which results in the reduced production of hexokinase, thus decreases the production of ATP in the mitochondria and glycolytic pathway, reducing the speed of the X sperm without affecting the speed of Y sperm. Sperm motility assay after treatment with agonists. Treatment with toll-like receptor 7 and 8 ligand R848 slows down X sperms with no effect on Y sperm. Upper layer sperm form 90% of the male embryos in both mice and cattle. Precipitated sperms form 80% of the female embryos. Computer-assisted sperm analysis showed shorter sperm tracks and decreased average path velocity. The slow motility was reversed by removing resiquimod with no effect on the acrosome status, sperm viability, and the fertilization ability. Conclusion. The only difference between X and Y spermatozoa lies in the DNA content. Recent proteomics and genomic studies have identified a set of proteins and genes that are differentially expressed between X and Y spermatozoa. Sperms can be separated by manipulating the functional differences due to the differential expression of genes and proteins. Future direction. The scientists are taking advantage of the toll-like receptor 7 and 8 in the immune system to design new treatments for the treatment of COVID-19 infection. The role of toll-like receptor 7 agonist imiquimod has been explored to find new treatment for the COVID-19 infection. However, further research needs to be done to see if COVID-19 will alter the motility of the sperm in the population and alter the sex ratio. Thank you and have a wonderful day.